In the last few chapters of our text, we've talked about genetics, and we've talked about how genes are passed on from parents to offspring. In this particular chapter, chapter 9, we're going to talk about some of the most basic ideas in biology. We're going to talk about how a gene is expressed. In other words, how that piece of information that's coded in your DNA actually comes to bring about the phenotype that we observe. We talked about all sorts of genes and how they are bring about a phenotype in the past few chapters. And so now in this chapter we want to talk about the details of how that actually occurs. It's an exciting chapter, and we should note that we know more at this point in history than we ever have known before about the expression of genes. And that leads us to, or allows us to do some really dramatic things that were only science fiction sort of ideas just as recent as a couple decades or even a decade ago. So at this point in history, we have the ability to take a gene from one organism and to place it into another organism, and that organism will express that particular gene. A great example is shown on the screen. We have a petri dish that has some glowing bacteria in it. This glowing bacteria is transgenic. It has a gene that has been taken from a jellyfish in the ocean that glows, and that gene has been placed into the bacteria, and the bacteria glow as a result of the insertion of that particular gene. It's an exciting topic, and this is relatively common in today's world, something that is not restricted to the most expensive and sophisticated laboratories. This fall, in our class on campus for majors, we'll be working with some of these transgenic bacteria that actually glow. And so this brings about both some really remarkable activities that we can accomplish, and it brings about some controversy. And so the other picture you see on your screen is of crops that are genetically modified. And so we have the ability today to take genes from one plant and place them into another plant, and that provides us some advantage for growing crops. For instance, a large percentage of the corn, over half the corn and soybeans that are grown in this country, are genetically modified. They are what we refer to as BT corn or BT soybeans. This means that a gene that's typically produced by a bacterium has been placed into these corn and soybeans. And so this gene produces a protein that kills insects. And so you would imagine that if you take this gene and place it into corn and soybeans, you have a plant that produces its own insecticide, which is great in many aspects because it reduces the amount of insecticide that you have to use. So it reduces costs for the farmers, and it reduces the exposure of everyone to those insecticides. And so the use of genetically modified organisms has this great potential to reduce our exposure to insecticides. There are lots of other things too, and so there's a project going on right now that they're working on inserting a gene for a nutrient into rice, and so a large percentage of the world suffers from a disorder of this particular nutrient. If they're successful in taking this gene and placing it into rice, that then literally millions of people that now suffer from a deficiency of this particular nutrient will have it as a result of the fact that it's incorporated into rice. There's also projects that are ongoing at the current time that are designed to make plants like corn less dependent on water. And so corn is a very water needy. It demands, in order to grow corn, it takes a lot of water. And so much of, for instance, the continent of Africa is unsuitable to the production of corn as a result of the fact that it's simply too dry. And so if corn can be genetically modified so that it can grow under more arid conditions, then this has the potential to bring food to large sections of the world that currently are malnourished. And so what I've just described are some very positive aspects of genetically modified crops. I should point out that genetically modified crops are very controversial, and it's not uncommon, as is depicted at the very beginning of your lecture here, for people to be against genetically modified organisms and to protest this so-called Frankenstein food or Frankenfood, food that has genes that have been transplanted from other organisms. And so there are downsides. There are dangers that are associated with genetically modified organisms. Some of these things include questions such as what happens if these genetically modified organisms either escape, which is not really an issue in the case of plants, but we have genetically modified animals too. But what happens if genetically modified plants cross with wild plants? What are the consequences of that? And that's a legitimate concern. What happens if the residue of these crops, so for instance the corn that produces its own insecticide, what happens when that makes its way into the environment? What impact does that have on organisms? And so these are all legitimate questions. And the purpose of our chapter is not to tell you that genetically modified organisms are the best thing in the world, but the purpose of our chapter is to provide you with an understanding of exactly what genetically modified organisms are, so that you can make up your own mind based on science, based on facts, and not just based on emotional testimony from people that do or do not like genetically modified organisms. So when we complete this chapter, you'll have a relatively thorough understanding of what genetically modified organisms are, and be able to make up your mind on what you think about them. It is interesting to note that much of what you consume today is the result of genetically modified organisms, simply because over half the corn and soybeans grown in this country are genetically modified. Alright, so let's move on. This is a great chapter, and we're going to start with this idea of protein synthesis. And so remember, this is an extremely important idea. As we pointed out before in our class, every single cell in your body has the exact same DNA. A hair cell, liver cell, a skin cell. The DNA is all exactly the same, but the key is this: it's expression of that DNA. Just a small subset of the DNA is expressed in a liver cell, and that's what makes that liver cell look like a liver cell. And the same thing applies to a skin cell, a brain cell, all these other cells. And so DNA expression is extremely important. And so protein synthesis then is the process of using the instructions carried on a gene to actually produce proteins. And we've talked about the idea in the past in our class that one protein codes for one gene, and the process of protein synthesis then is the process of expressing that gene. There are several steps that involved, and both of these require, or these together require, both DNA and RNA. So we're familiar with the definition of a gene. A gene is a short sequence of DNA that encodes a protein, and then we're familiar, of course, with what a protein is, a long chain of amino acids. We've known that for some time. We learned about those things in a very early chapter in our class. So, excitingly, here is the most basic idea in all of biology. We refer to this as the central dogma of biology. You have to understand this to understand anything about the living world. As you drive down the road and look around you at the trees and the animals, the grass that surrounds you, this process is taking place, and this is the underlying theme that causes everything to look and appear the way that it does. And so the central dogma of biology is this: that everything starts out as DNA. DNA is turned into RNA. We're at the top of our slide here now. And then RNA is turned into a protein. And so we go from DNA to RNA to protein. There are two processes that we utilize to make this transformation. Going from DNA to RNA is a process that we refer to as transcription. Going from RNA to protein is a process we refer to as translation. We'll talk first about transcription and then about translation. So remember, the amazing idea in biology is that all the information to produce an organism is stored in the DNA. When you look at a cow or a piece of grass or a snake or a dinosaur, all of these things look the way they look because of this information that's encoded in the DNA. You, as a human, appear the way that you appear as a result of the information in the DNA. I have red hair. Other people have dark hair. Some people have blonde hair. And all of this phenotype is a result of the information encoded in your DNA. Now, as a brief review, something we've talked about many, many times in the course of our class, we know that DNA is a double-stranded helix. And we know that the outside of the ladder of the molecule, as covered in the last unit of our chapter of our text, our class, actually the first unit of our class as well, The outside, or the handrails of that ladder, are composed of sugars, shown in red, and phosphates, shown in purple. And the rungs of the ladder are composed of complementary nucleotides that are held together by hydrogen bonds. And so no doubt you remember that from earlier in our class. It's fascinating to note at this point that the information is found in the bases, as you realize. These bases that project forward towards the center of the DNA molecule, thymine for instance. So everything about you is written out in this order of bases. Everything about every living organism is written out in this order of bases that's found in the DNA. So the DNA is going to be transferred to RNA. In this process of expressing DNA, we first have to transfer the information in DNA to RNA. RNA is a similar molecule, but a little bit different. It's also a nucleotide. It's also made of nucleotides. The molecule itself is a nucleic acid. And RNA, interestingly, is single-stranded. So this is RNA shown in our slide here, not double-stranded. RNA also has a backbone of sugars and phosphates, and it also has bases that are attached to that sugar-phosphate backbone. There's one difference in the bases that we find in RNA versus the bases in DNA. In DNA, we have the base thymine. In RNA, we have the base uracil that replaces it, that's shown right here. So all the other bases are the same, though. We still have 4 in RNA. In DNA, we had thymine, adenine, cytosine, and guanine. In RNA, we have uracil, and then those other 3 bases as well: cytosine, adenine, and guanine. So remember, the one difference in bases that we see in RNA, and you can see that here, guanine, adenine, guanine, uracil. The one difference in bases in RNA is that we have uracil instead of thymine, which is going to be important in just a moment. Again, as a review from a moment ago, there are 2 steps in going from gene to protein, the central dogma in biology. Transcription, in which we go from DNA to RNA; translation, in which we go from RNA to protein. Here's transcription. We're going to talk about this first, and then we'll talk about translation in the following video. Transcription takes place in the nucleus. We realize the DNA is located in the nucleus, and transcription takes place when an RNA polymerase comes along and binds to a promoter region of the DNA. And so a promoter region is a region that's found just before a gene. Each gene has a promoter region. The RNA polymerase binds to that region, and then the RNA polymerase moves forward. And as it moves forward, it unzips the DNA. You may recall that in DNA replication, DNA helicase performed this function. But in the case of RNA production in translation, RNA polymerase unzips the DNA. And here's what the RNA polymerase does. It takes a piece of the DNA. It only reads one side of the DNA, which is an interesting idea, an interesting concept, and something we need to remember. So the 2 strands of DNA are separated, and one side is read. And what's happening here is that the RNA polymerase is producing RNA based on the sequence of bases it finds in the DNA. So every time it sees an adenine here, pretend that I'm pointing to an adenine with the pointer here in the DNA. If it was DNA, it would lay down a thymine, but since it's RNA, it lays down a uracil. If this is a guanine in the DNA, then a cytosine is laid down in the RNA. If this is a thymine in the DNA, then an adenine is laid down in the RNA. And so this idea of complementary base pairing holds and is still true for the production of RNA. And so the RNA polymerase moves down the DNA. But one key difference here is this. If we look at the difference between DNA replication and the production of RNA in transcription, and the difference is this, that only a small section of the DNA is going to be read and produced by the RNA polymerase. And so typically one gene or just a couple genes at a time are going to be read. This is different from DNA replication, because in DNA replication the entire chromosome is read in this manner. And so remember, this is the first step in the production of proteins from your DNA. Here we see a video of this occurring. And so the blue molecule that we see is the RNA polymerase. It is bound to a promoter, and it is moving down the DNA, separating the 2 strands of the DNA molecule. And so now the RNA polymerase is moving down the DNA and laying down complementary RNA as it comes across the DNA bases. And so, of course, this molecule is the RNA, and then the bases shown here are the bases found in DNA. Let's let it continue. Now, interestingly, I believe what we'll be able to see here in a minute is that the RNA is trailing off of this little section that we refer to as a transcription region. The DNA has been separated in a very localized region, and it's going to re-zip as this RNA polymerase moves along. And watch, this RNA that's being produced is going to just hang out of this particular region. So if we zoom back out a little bit, so there we see the RNA molecule that's being produced. We see the DNA re-zipping and the RNA polymerase moving right along through that DNA, producing RNA from the DNA in the process, via the process of transcription. Eventually, here we see, there we see the end of it there. Eventually, when the RNA polymerase reaches the end of the gene, the RNA polymerase is going to leave, the DNA zips up entirely, and the RNA molecule leaves and is going to leave the nucleus and go to the next step, which is what we're going to see in the next video, is the process of translation. So remember that this process is extremely important and is happening right now in millions of cells of your body. In order for you to survive and for your normal physiology to occur, all of your cells are going through this process. Any living cell in your body is producing proteins at this time. So what we just observed is happening in your body in millions of instances at this particular moment. In the next video, we'll look at what happens to this RNA molecule as it goes on to complete this process of becoming a protein.